Welcome back to the deep dive. When we think about genetic diseases, we often picture maybe a gene that's broken or missing entirely, or maybe a simple typo in the DNA code.
Right. But today we're looking at something, well, fundamentally different, repeat expansion disorders.
Exactly. These aren't about missing information, they're about too much information. It's like a genetic stutter, right? A
Yeah.
tiny DNA sequence just repeats over and over and over again.
And it's when that number of repeats crosses a certain threshold that things go wrong, it becomes pathogenic. The sheer number of copies causes the problem.
So our goal today is to really unpack two key examples, Huntington's disease and Fragile X syndrome. We want to see how the location of that stutter dramatically changes the disease.
And also look at the specialized tools needed to even detect these unstable repeats. We've pulled together insights from quite a few high-level genetics sources, focusing on how these are diagnosed and the mechanisms behind them.
Okay, let's dive in. Case one. Imagine a man, let's call him James, he's 45, started noticing, memory issues, trouble concentrating,
Yeah.
but then it got more serious. Involuntary movements, sort of jerky motions, chorea, I think it's called, facial grimacing, plus big emotional shifts, anxiety, bad sleep.
Yeah, that combination, the cognitive decline, the behavioral changes, and especially the chorea that really points strongly towards Huntington's disease,
Right.
HD.
Which is pretty serious, right?
Oh, absolutely. It's a fatal neurodegenerative disorder. It's autosomal dominant, meaning you only need one copy of the faulty gene. And that onset, 30s to 50s, is very typical.
And if you were to look inside the brain, say with an MRT scan,
You'd see something quite specific. There's significant atrophy, shrinkage, particularly in a part of the brain called the striatum. That includes the putamen and the caudate nucleus.
areas involved in movement and emotion.
Exactly. You see a very targeted loss of neurons there, which directly connects to the symptoms James is experiencing. The ventricles might look enlarged, too, because of the surrounding tissue loss.
Now, here's a really interesting part from the sources, the history and geography. Huntington's isn't common everywhere, but there's this incredibly high prevalence near Lake Maracaibo in Venezuela.
Ah, yes. A
Right.
classic example of the founder effect.
Right. Traced back, apparently, to just one woman in the 19th century. Dr. Nancy Wexler's work there was groundbreaking, starting back in the 70s.
It's a perfect illustration. A small group splits off, maybe just a few families. By chance, one founder carries a rare gene variant, in this case the HD mutation. In that isolated population with limited gene flow from outside, that rare allele can become much, much more common over generations than it would be in the general population. It's population genetics in a tragic natural experiment.
So we know where the damage is, those neurons in the striatum. What's the actual genetic fault causing this?
It all comes down to a specific repeat, CAG. It's a trinucleotide repeat, meaning 3 DNA letters, C, A, G, repeated over and over.
Okay.
And crucially, this repeat sequence is located right inside a gene in exon 1 of the HTT gene on chromosome 4.
And that CAG codes for?
Glutamine, an amino acid. So when the gene is translated into the Huntington protein, this CAG expansion creates a long, sticky tail of glutamine residues, a polyglutamine tract.
Is there like a magic number? How many repeats are okay, and when does it become a problem?
There are pretty clear thresholds, actually. If you have 26 CAG repeats or fewer, you're generally in the clear. That's considered normal.
Okay.
But once you hit 40 repeats or more, that's considered full penetrance. The disease is essentially guaranteed to develop at some point.
What about the space in between 26 and 40?
That's the intermediate or reduced penetrance range, roughly 36 to 39 repeats. People in this range might develop HD, often later in life, but it's not certain.
And there's a direct link between the number and how bad the disease is.
Absolutely. Critical point. More repeats generally means an earlier age of onset and a more severe, rapidly progressing disease. We see this tragically in juvenile HD, where individuals often inherit 60 or even more repeats. There's a clear inverse correlation, more repeats, younger onset.
So this mutant Huntington protein, the one with the extra long glutamine tail, what's it actually doing wrong inside those neurons? Why is it toxic?
The core problem is that the mutant protein, MHTT, doesn't fold correctly. It misfolds. And misfolded proteins tend to be sticky.
Sticky out.
They clump together, forming these toxic aggregates or inclusions inside the neuron. You can think of it like, like accumulating gunk or sludge inside the cell machinery.
And that gums everything up.
Pretty much. It interferes with all sorts of vital processes. For one, it disrupts transcription condition the reading of other genes. For example, it reduces the production of important survival factors for neurons, like BDNF.
Okay, brain-derived neurotrophic factor.
Right. It also impairs the cell's garbage disposal systems, the proteasome and autophagy, which normally clear out damaged proteins, so the toxic stuff builds up even more.
Vicious cycle.
Exactly. And it messes with mitochondrial function, the cell's powerhouses. Eventually, the cumulative stress is just too much for the neuron, especially those vulnerable ones in the striatum.
Okay, so that's the what. What about the how? How do these repeats actually get longer from one generation to the next? You mentioned this instability.
Yeah, the mechanism behind the expansion is thought to be replication slippage. It happens during DNA copying.
When the cell divides.
Precisely. Especially in these repetitive DNA regions, the new strand being synthesized can kind of slip or misalign relative to the template strand. It can loop out.
Like a little bulge.
Kind of. And the DNA copying machinery can then mistakenly copy that looped out section, effectively adding extra repeat units into the new DNA strand.
And that happens generation after generation.
It can. And that inherent instability, that tendency for the repeat number to increase, is what leads to anticipation.
Which is the phenomenon where the disease appears earlier and often more severely in successive generations.
Exactly. And interestingly, in Huntington's, this expansion is more likely to happen, or happen to a greater degree, when the gene is passed down from the father. Paternal transmission often leads to larger jumps in repeat number.
That brings up a crucial point you mentioned earlier. Location, location, location. HD's CAG repeat is in an exon, the coding part of the gene.
Right, and that leads to a toxic protein. But that's just one way these repeats can cause trouble. Where the repeat occurs dictates the entire disease mechanism.
So what are the other possibilities?
Well, if the repeat expansion happens in a non-coding region, like the untranslated regions, the UTRs at the 5' or 3' end of a gene.
The bits before or after the actual protein recipe.
Exactly. Or even within an intron, one of those intervening sequences. The effect is totally different. Instead of making a toxic protein, you might interfere with the gene's expression itself.
Can you give us a quick rundown?
Sure. So exonic repeats, like HD, make toxic proteins. Repeats in the 5A or UTR, as we'll see with Fragile X, often lead to the gene being silenced, turned off completely.
Okay.
Repeats in introns can sometimes block the whole process of transcription, like in Friedreich ataxia. And repeats in the 3 or UTR can create toxic RNA molecules that trap other important proteins, which is what happens in myotonic dystrophy.
Different location, different mechanism entirely. Let's pivot to that 5' UTR example now.
Yeah.
Case 2. Andrew, 6-year-old boy.
Okay.
He presents with intellectual disabilities, hyperactive, has some speech and motor skill delays. But unlike James, he hasn't lost skills, he's just generally delayed. And crucially, there's a family history. His mom and aunt and uncle all have some learning or intellectual disabilities.
This sounds very much like Fragile X syndrome, or FXS. It's actually the most common inherited cause of intellectual disability, particularly in males because the gene is on the X chromosome.
And besides the developmental delay,
You often see characteristic features, though not always. Things like a long face, large ears, maybe macrocephaly, a larger head size. In boys after puberty, macroorchidism is common. Behaviorally, you might see hand flapping, hyperactivity, sometimes autistic features, or tantrums.
and the genetics here involve a different repeat, right? Not CAG.
Correct. FXS is caused by an expansion of a CGG repeat. And critically, it's located in the phi UTR of the FMR1 gene on the X chromosome, right before the coding sequence starts.
And again, there are thresholds.
Yes, very distinct ranges. Normal is fewer than 44 CGG repeats. Then there's an intermediate or gray zone allele, roughly 45 to 54. Then the premutation range, which is 55 up to 200 repeats. And finally, the full mutation, which is anything over 200 repeats.
Okay, let's focus on the full mutation first, the 200 repeats that cause classic Fragile X syndrome in Andrew. How does that work? It's not a toxic protein like HD.
No, completely different. It's a loss of function mechanism. When you get that massive CGG expansion in the 5' UTR, something dramatic happens chemically. The promoter region of the FMR1 gene gets hypermethylated.
Methylation, like silencing tags on the DNA.
Exactly. Think of it like pouring concrete over the gene's on switch. This heavy methylation shuts down transcription completely. No messenger RNA is made from the gene.
So no FMRP protein.
Correct. There's a deficiency or complete absence of the FMRP protein, a Fragile X mental retardation protein.
And what does FMRP normally do? Why is losing it so bad for brain development?
FMRP is really important. It's an RNA binding protein that acts like a brake or a regulator for protein synthesis right at the synapse, the connection point between neurons. It helps control the local production of specific proteins needed for synaptic function and plasticity.
Mm.
When FMRP is missing, that brake is gone. Protein synthesis goes unchecked.
Temporism.
It leads to structural problems like immature, spindly dendritic spines on the neurons and impaired signaling between them. This fundamentally disrupts brain development and learning, causing the intellectual disability and other features of FXS.
Okay, loss of function for the full mutation. But what if the pre-mutation range 55 to 200 repeats? Those individuals don't usually have classic FXS, but they can get other problems, right?
Yes, and this is where it gets even more complex, because the mechanism shifts again. The premutation doesn't cause disease through loss of FMRP. In fact, people with the premutation often have normal or only slightly reduced FMRP levels.
So what's the problem then?
It seems to be mainly RNA toxicity. In the premutation range, the FMR1 gene is actually transcribed more actively. You get an overabundance of the messenger RNA containing that expanded CGG repeat tract.
And that excess RNA is bad.
Yes. The long CGG repeat sequence in the RNA causes it to fold up into abnormal hairpin structures. These RNA hairpins act like sticky traps.
Traps for what?
They sequester or bind up various other important RNA-binding proteins within the cell, pulling them away from their normal jobs. This disruption of cellular protein machinery and RNA metabolism is thought to underlie conditions associated with the premutation, like affects fragile X-associated tremor ataxia syndrome. This typically affects older carriers, especially males, causing tremor, balance problems, and cognitive decline. Totally different symptoms from FXS itself.
Wow. And also the premature ovarian failure in women.
Exactly. Premature ovarian failure, or POF, is another condition linked to the premutation in female carriers. Again, likely driven by this RNA toxicity mechanism, though the exact details are still being worked out. And of course, premutation alleles are unstable and can expand to full mutations when passed down, especially from the mother. So anticipation happens here too, but via maternal transmission.
So, two diseases, HD and FXS, both caused by repeat expansions, but radically different locations, mechanisms, and outcomes. This must present a real challenge for diagnostics. How do you actually measure these repeats, especially when they can get so huge and unstable?
It's definitely not straightforward with standard methods. You need specialized techniques. And interestingly, one of the older techniques, the Southern blot, is still incredibly valuable, particularly for the very large expansions.
Southern blot, that's one involving gels and probes, right?
Yeah, it's a bit labor-intensive. You cut the patient's DNA with specific restriction enzymes, then run the fragments out on a gel to separate them by size. Then you transfer the DNA pattern to a membrane and use a labeled probe that sticks only to the gene region you're interested in.
How does that help with, say, Fragile X?
For FXS, the Southern blot is crucial because you can combine it with methylation-sensitive enzymes. You use an enzyme like EGI that only cuts DNA if it's not methylated.
So it checks the silencing status, too.
Precisely. In a male with a full mutation, 200 repeats, the FMR1 promoter is hypermethylated. So EGI cannot cut there. What you see on the blot is just one large DNA fragment, typically bigger than 5.2 kilobases, representing the expanded, methylated, inactive allele. It confirms both the large size and the silencing.
Very clever, but it sounds slow. Is there a faster way, maybe for screening?
Absolutely. The go-to method for screening and for sizing smaller expansions now is triplet primed PCR, or TPPCR.
TCR polymerase chain reaction, but triplet primed.
It's a specialized PCR setup. It uses 3 primers. 1 forward primer outside the repeat, 1 reverse primer outside the repeat, and a 3rd primer that actually binds within the repeat sequence itself, but only anneals efficiently when there's an expansion.
Okay, and what does the result look like?
Instead of getting just 1 or 2 clean peaks showing the allele sizes, if there's a large expansion, the TPPCR generates a very characteristic pattern on the electropherogram output. It looks like a ladder or a stutter.
A stutter?
Yeah, a series of peaks, each separated by the size of 1 triplet repeat. This stutter pattern is a clear visual indicator that a large, unstable repeat expansion is present.
So for HD, you might see 1 normal peak, say at 12 repeats, and then this long, trailing stutter pattern going way out.
Exactly. It tells you immediately that there's a normal allele and a significantly expanded, potentially pathogenic allele. It's much faster and uses less DNA than a Southern blot, making it great for initial testing. Though Southern might still be needed for precise sizing of huge alleles or confirming methylation in FXS.
So we've covered a lot of ground here. Huntington's, Fragile X. These dynamic mutations are really fascinating.
They really are. I think the key takeaway is just how critical the location of that repeat is.
Hmm.
An exon expansion like HD gives you a toxic protein. A 5-foot UTR expansion like FXS gives you gene silencing and loss of function, or RNA toxicity in the premutation.
Different mechanisms, different diseases, all from a DNA stutter.
Right. And we need those specialized tools, the Southern blot and TPPCR, to grapple with the instability in size and sometimes the epigenetic changes like methylation that define these disorders.
Okay, let's wrap up with a final thought for our listeners to consider. We've seen these vastly different diseases, a movement disorder like Huntington's, a developmental disorder like Fragile X, yet they both stem from the same fundamental issue. Our DNA's own inherent instability when these little repetitive sequences get too long.
Mm-hmm.
So thinking about things like replication slippage and anticipation, could understanding and maybe even targeting the instability itself, the process that makes these repeats grow, be a future therapeutic avenue,
Mm-hmm.
rather than just dealing with the downstream consequences like the toxic protein or the missing protein? What if we could stabilize the DNA structure itself?
That's certainly a compelling direction for future research, a really provocative question to end on.